The adaptive component of our immune system employs several types of lymphocytes. The function of one type, called B cells, is to release antibodies that can bind to specific antigens. Antibodies perform a number of roles, including coating and thereby neutralizing foreign invaders, preventing them from entering or damaging host cells. The antibodies also flag the invaders, making them easier for macrophages to engulf and destroy. The function of another type of lymphocyte, called cytotoxic T cells, is to bind to and destroy infected host cells. Cytotoxic T cells use their T cell receptors to recognize foreign antigens displayed on host proteins, called MHC proteins. By destroying infected cells, cytotoxic T cells limit the infection in the body. Because antibodies are in large part found in the bloodstream, which is considered a body humor, antibody-based responses are referred to as humoral immunity. Responses by cytotoxic T cells are called cell-mediated immunity. Neither B cells nor T cells can function, however, without the help of a third type of lymphocyte called helper T cells. Helper T cells release molecules called cytokines that are required to activate B cells and cytotoxic T cells. The activation and differentiation of helper T cells determines whether the immune system delivers primarily a humoral or cell-mediated response. Helper T cells begin as naive, undifferentiated cells called Th0 cells that need activation by an antigen-presenting cell, or APC, that has processed antigens from a microbial invader. An antigen-presenting cell engulfs microbial invaders or their cellular debris, breaks down the foreign material within the cell, and then puts small peptides from the microbes onto the cell's own proteins, called major histocompatibility, or MHC2 proteins. A helper T cell has T cell receptors that may be able to bind to the antigen MHC2 complex. A co-receptor called CD4 participates in binding to MHC2. The binding to MHC2 provides naive helper T cells with their first activation signal. A protein called CD28 on the T cell surface binds to a protein called B7 on the surface of the antigen-presenting cell, and this binding provides the second required signal for activation. Different invaders influence how Th0 cells mature. The differentiation into either Th1 or Th2 cells represents two main pathways. Mature Th1 cells perform a number of tasks. They stimulate macrophages to kill intracellular microbes, stimulate cytotoxic T cells to kill infected cells, and also stimulate some B cell activity. In contrast, Th2 cells primarily activate B cells. Thus, the Th1 response is set up to attack intracellular pathogens, while the Th2 response attacks extracellular pathogens. If the initial invader is a bacterium, the antigen-presenting cell may recognize bacterial products, such as components of the cell wall. In response, the antigen-presenting cell releases a cytokine called IL-12, which stimulates Th0 cells to differentiate into Th1 cells. Parasitic worms are strong inducers of the Th2 pathway. The process of Th2 induction is not clear, however. Products from the invader may stimulate antigen-presenting cells. However, it's also possible that other cells present near the infection, such as mast cells, release a cytokine such as IL-4, triggering Th2 differentiation. Although much is yet unknown, it is clear that the parasites themselves play a large role in the decision between these two pathways. The effector function or purpose of Th1 and Th2 cells is to release specific sets of cytokines. By releasing one set of cytokines, Th1 cells can stimulate cytotoxic T cells to kill pathogen-infected cells. As shown earlier, the first signal needed to activate cytotoxic T cells is the binding of the T cell receptor to an antigen MHC complex, in this case MHC class I, on an antigen-presenting cell. Activated Th1 cells then provide the second signal, which includes cytokine IL-2. Upon binding of IL-2, the cytotoxic T cell replicates and gains cytotoxic activity. The cells travel to the site of infection and kill any cell bearing the same peptide MHC class I complex as presented to them by the antigen-presenting cell. Th1 cells can also activate macrophages harboring intracellular bacteria. To activate the macrophage, the Th1 cell's T cell receptor binds to an MHC2 antigen complex and secretes interferon gamma. The activation helps the macrophages kill the intracellular bacteria. As shown in these examples of Th1, the effector function of helper T cells is to produce cytokines. Th1 produces IL-2 and interferon gamma. Th2 cells produce IL-4, IL-5, IL-6, and IL-10. Although the cytokines have their own functions in activating the immune system cells, they also help enforce the initial decision between the Th1 and Th2 pathways. Interferon gamma from Th1 inhibits Th2 development. Likewise, IL-4 and IL-10 production from Th2 inhibits Th1 development. The primary activity of Th2 cells is to release cytokines that activate B cells. Immature B cells produce a specific antibody on their cell surface. The cross-linking of the antibody with antigens provides the first signal for B cell activation. B cells are a kind of antigen-presenting cell because they can internalize antigens, process the antigens, and display them on MHC2 proteins. If an activated Th2 cell recognizes the antigen, it will bind to it. The binding allows the interaction between two proteins on the T and B cells called CD154 and CD40, triggering the second intracellular signal needed for B cell activation. Th2 also releases B cell stimulating cytokines. IL-4 stimulates the B cells to proliferate. IL-6 stimulates the B cells to differentiate into antibody-secreting plasma cells.